In the first session of meninges, we have discussed the layout of the meninges in the brain and spinal cord in the form of three layers: dura, arachnoid, and pia mater. We have discussed the dura mater in detail, and we have discussed the sinuses, venous sinuses, dural venous sinuses, which are formed between the periosteal and the meningeal layers of the dura. Now we are going to focus on the spaces which are formed between these three layers, and we are going to discuss the arachnoid matter and the pia mater in detail. At the end, we are going to discuss the cisterns which are formed between the arachnoid and the pia mater. The spaces that are found between the three layers of the meninges that we need to identify are the epidural space, this space is found between the dura mater and the skull bone. Between the dura and the arachnoid, we have the subdural space. The word sub means beneath, so this layer is present beneath the dura and above the arachnoid. The third space is the subarachnoid space, and this space is present between the arachnoid mater and the pia mater. Now this space is important because it contains CSF, which is the cerebrospinal fluid. We will look at this illustration to have a better understanding. In this picture, we need to identify the skull bone, and then in the pink color we can see the two layers of the dura mater. In the dark pink color we have the arachnoid mater, and the purple one refers to the pia mater. So the space which is present between this dura mater and the arachnoid mater, this space is the subdural space, which contains a thin film of fluid, while the space which is present between the arachnoid mater and the pia mater, the important one, this space will be the subarachnoid space, which will contain our cerebrospinal fluid. Now we are going to identify the second layer of the meninges, which is the arachnoid mater. This layer is defined as a thin impermeable layer of membrane which is present just beneath the meningeal layer of the dura. So here in this pale skin tone, the layer that we identified is known as the arachnoid mater. Important features of this layer are that it has certain projections identified as the arachnoid villi, which are numerous in the area of the brain, and they are projected into the dural venous sinuses. So these projections that we see going from the arachnoid to the dural venous sinuses, these are identified as the arachnoid villi. Aggregates of these arachnoid villi become the arachnoid granulations, and these are the important sites for the CSF absorption. So to maintain the constant pressure of the cerebrospinal fluid, these arachnoid villi and the arachnoid granulations are important. All the cerebral arteries and the cerebral veins, they are also present in this layer. And other important feature of this arachnoid matter is that it sends a sleeve around the second cranial nerve, which is the optic nerve. By sending a sleeve around the cranial nerve too, the subarachnoid space is extended as far forward to the eyeball as the optic nerve extends. So the arachnoid matter important features are the presence of the arachnoid villi and arachnoid granulations, and number two, it sends a sleeve around the cranial nerve, number two, which is the optic nerve, by which the subarachnoid space is extended to the back of the eyeball. Moving on to the third layer of meninges around the brain and spinal cord, which is the pia mater. This is a thin layer which is vascular in nature, and it closely invests the surface of the brain, specifically in the regions of the sulci and gyri, where it dips deep. This layer extends into the cranial nerves, around the cranial nerves, and it merges with the epineurium, which is the outer layer of the nerves, and it fuses with the epineurium. The important fact about this vascular layer is that it forms a very important vascular structure which is known as the tela choroidea. So here is the picture where we can see the lateral ventricles. This is the coronal section explaining the ventricle system of the brain. This is the lateral ventricle, this is the third ventricle, and here the pia mater has formed this vascular structure which is known as the tela choroidea. Now this tela choroidea is very important because this tela choroidea along with the ependyma, it merges and it forms another very important structure which is known as the choroid plexus. So this choroid plexus is actually formed by the tela choroidea, which is contributed from the pia mater and the ependyma. So this is the tela choroidea, and this fuses with the ependyma here to form the choroid plexus in the roof of the lateral ventricles, and here we can see the choroid plexus formed in the roof of the third ventricle. The significance of this choroid plexus is that it is present in the roof of third ventricle, lateral ventricles, and also the fourth ventricle, and this is the side where the CSF is produced. So now we can say that for the CSF production, pia mater is important because it produces the choroid plexus, and for the absorption of the CSF we have arachnoid matter, which forms the arachnoid villi and granulations projecting into the dural venous sinuses. The last concept that we need to develop is the cisterns. Cisterns are actually the CSF filled enlarged spaces which are formed between the arachnoid and the pia mater, that is between the second and third layers of the meninges. Most of the cisterns are appreciable in the areas of the brain and around the brainstem. However, there is one cistern which is identified in the area of the spinal cord. So to name few of them, the important one is the cisterna magna. This is the largest of all the cisterns. The other name for this cistern is cerebellomedullary cistern. The second one is the pontine cistern, which is in relationship to the pons area. Another cistern which is related to the cerebral peduncles of the midbrain is the interpeduncular cistern. Another example is the cisterna quadrigemina, which is in relationship with the cerebellum and the corpus callosum. And then there is one cistern which is found in the area of the spinal cord, which is known as the lumbar cistern. These are only the few examples. There are a number of cisterns. So let's look into the pictures to have a better understanding of these cisterns. The first cistern that we need to identify here is this one. This space filled with the cerebrospinal fluid, which is found between the medulla and the cerebellum. This is identified as the cerebellomedullary cistern. The other name for which is cisterna magna, as this is the largest of all the cisterns. And the second one is the cisterna pontis, or the pontine cistern, which is present in the anterior surface, or which is present on the anterior surface of the pons. Remember, this is the side view in which we can appreciate the brainstem, corpus callosum, and the area of the cerebellum. The third cistern is cisterna interpeduncularis, which is present here between the two cerebral peduncles of the midbrain. And the fourth cistern here is the cisterna quadrigemina, which is in relationship to the splenium of the corpus callosum and the cerebellum. This is the site of the lumbar cistern, which is present at the end of the spinal cord, and this one, and it extends from the conus medullaris all the way to the S2 vertebrae. So the cisterns are nothing but the spaces filled with CSF between the arachnoid and the pia mater, and we have identified few of them as cisterna magna, pontine, interpeduncular, and the cisterna quadrigemina. The one in the spinal cord that we have just seen is the lumbar cistern. That's all about the 3 layers of the meninges. In the next video, I am going to briefly summarize the difference of meninges which are found between the brain and the spinal cord area.